Welcome to the series in Food Microbiology. In this series of lectures, in the last couple of sessions, we were seeing the different methods to identify and detect the presence of microbes in food. In this session, we are going to talk about two major aspects. One is the detection of bacterial toxins, and two, a rapid and a fast method for the detection of pathogens. In many cases, even the presence of microbe is not required for a food to cause a foodborne illness. It is enough if the microbial toxins, that is the microbial metabolites, are present. So even if the microbe is not there, if the microbial toxins are present in the food, that can cause major episode of food poisoning. So how do we detect that? What are the different methods? Because so far whatever method we saw, it was to detect the presence of the microbe, the living organism. But here we are going to talk about how to detect the presence of bacterial toxins, and then which are the rapid methods in how fast can we detect the presence of the microbes, what are the methods which can be used for that. Test for the bacterial toxins in the food. Staphylococcus aureus, Clostridium botulinum strains and several others like Bacillus cereus, the Vibrio parahemolyticus, all these organisms while growing in the food can produce toxin and cause intoxication or food poisoning among the consumers. So it is not going to be the organism which is actually lethal. It is the toxin produced and secreted by the organism which remains in the food which is going to cause the foodborne illness. So we need to test not only for the presence of the organism but also for the presence of the toxins. Specific methods have been developed to test for the presence of these toxins. For example, to detect the Staphylococcus aureus enterotoxin in foods, the recommended method is to initially extract the toxin from 100 grams of food and then to concentrate the toxin in 0.2 ml sterile water or 0.2 molar saline. The presence of the toxin in the concentrated extract is then assayed against specific antibody by the microslide precipitation method. So this is a typical immunological method by which you extract the toxin, that is, the toxin is going to be a protein. So you extract, typical protein extraction method, I concentrate it, then resuspend it in either sterile water or saline, and then assay what is the concentration of this in the whole food. For example, you isolated so much nanogram or so much milligram from 100 grams of food, then whether you have to identify it using a specific antibody. So this is a typical immunological method, and this method is sensitive to 0.05 milligram of enterotoxin per ml of the extract. And in the case of Staphylococcus aureus, food poisoning can probably occur from consuming toxin less than 1 milligram of Staphylococcus. So if your food, for example, you isolated 0.5 milligram from 100 grams, it means that the concentration of the toxin in the food becomes very high, because the presence of 1 milligram is enough to cause food poisoning. So the level of toxin here becomes very important. We have other methods like radioimmunoassay, enzyme linked immunosorbent assay or ELISA, the reverse passive lactate agglutination assay or the RPLA. These can also be used in the place of microslide precipitation. To detect botulin, the toxin from Clostridium botulinum, the recommended procedure is used to extract toxin from the food. So like you extracted staphylococcal enterotoxin, the botulinum toxin is going to be extracted. It is a protein, so you have typical protein extraction method. Then to activate the toxin of non-proteolytic types, type B and E, a portion of the extract is then treated with trypsin. So we have two types like proteolytic and non-proteolytic types of toxin. So to activate the toxin, we treat it with trypsin. So trypsinized and non-trypsinized extracts in 0.5 ml portions are then injected. So once the protein becomes activated, then it is injected into mice intraperitoneally, and for each we take 2 mice per sample. Heated 100 degrees centigrade for 10 minutes, extracts are injected similarly, so that would be the control ones. So it is not, the control ones are going to be just heated ones, not trypsinized ones. Then the mice are then observed up to 48 hours for botulism symptoms and death. Typical botulism symptoms in mice would be ruffling of the fur, rapid and gasping breathing, weakness of the limbs and death due to respiratory failure. Even if the mouse is not dead, even if it shows even symptoms of ruffling and or gasping breathing, from which it can recover later on, it is enough that it shows the presence of botulinus toxin. The food is not safe to be consumed. Let's see the rapid methods to detect the microbes. The traditional or conventional methods used for the quantitative or the qualitative detection microbes in food and toxins, all these take a very long time. For example, when I talked about finding about the potability of drinking water, or in all those cases, in any wherever you are going to talk about the detection of microbes, you have to take then serial dilution, then put it for incubation. Depending upon the growth rate of the organism, you have to incubate it for 12 hours or 24 hours or 48 hours. So, which is actually a long time. So, other than going for this, do we have any rapid technology with which you can detect, so that within a matter of hours, you can say yes, the food is going to be rejected, or no, the food can be consumed safely. So, we have different rapid methods. Many of them are automated, have been developed to detect microbial loads, foodborne pathogens and their toxins. The first among these are the immunoassays. Several rapid and automated methods have been developed that rely on the specific antigen-antibody reaction and production of agglutination, color formation from chromogenic substrate, formation of an immunoband or fluorescence. So, immunoassays are based on antigen-antibody reaction. For example, like your blood grouping, a typical immunological method which depends upon the protein-protein interaction. So, antigen-antibody reaction, or you have the clot formation, agglutination, or when the food component reacts, if it contains a particular toxin, you can have a colorimetric chromogenic substance which can be broken down, indicating color production. So, the presence of the color or the absence of the color can indicate the presence of the pathogen or the toxin concerned, or formation of a band because of a substrate malignant formation, a complex formation, or presence of a fluorescence. The first among these is the immunofluorescence method. Commercially available specific fluorescence conjugated antibodies against the somatic or flagellar antigens of the pathogen are mixed with an enriched medium suspected to contain a specific pathogen like Salmonella on a glass slide. So, here what we do is we take a wash from the food stuff. We suspect that this wash contains the pathogen like Salmonella, and you enrich it, because even if the amount of pathogen present in the food material is less, you can enrich it and grow the organism. Now, what will we do is we have, for the detection of a particular pathogen, we will commercially raise antibodies, most probably against either some cell wall components or the flagellar materials of the pathogen concerned. So, these fluorescence conjugated antibodies would be mixed with the enriched medium. Now, when they following incubation and the removal of reagents, then you can now examine the microscopic slide under a fluorescence microscope, showing the cells showing fluorescence on the cell wall or the flagella or both. So, depending upon what type of conjugated antibodies we have using against a somatic or a cell wall component, or against we are talking about a flagellar antigen, if the pathogen is present, you will now be able to observe the fluorescence on the microscopic slide, which will indicate, which gives a visual observation that yes, the organism is present there. Direct fluorescent antibody techniques. These are fluorescently labeled monoclonal antibodies to bind and illuminate a target antigen. This is very rapid. For example, the picture shows against an antibody, fluorescent labeled antibody against the Legionella, a bacteria. This is not a food pathogen, but this is still a human pathogen. The fluorescent antibodies bind to the bacteria on the microscopic slide, allowing rapid detection of the bacteria using fluorescence microscope. So, when you mix the fluorescence antibody along with the microbial solution and observe it under the microscopic slide, this will go and bind either with the cell wall or the flagellar component, indicating the fluorescence would be highlighted using under the microscope. This is very valuable for visualizing certain bacteria which are difficult to isolate or culture from the sample. If the bacteria is very finicky, or it is very less in number, so that isolation becomes very difficult, or it is very difficult for isolation, even such samples can be detected using fluorescent antibodies. The RPLA method or reversed passive latex agglutination method. This has actually been developed to detect the toxins of several foodborne pathogens like Staphylococcus aureus, Clostridium perfringens, Bacillus cereus, etc. So, here with RPLA, we are not detecting the microbe, but we are going to detect the presence of microbial toxin. So, here what we do is soluble antigens such as the bacterial toxins will agglutinate in the presence of suitable antibody. That is what we are trying to do here. So, the antibody of a specific toxin is immobilized on latex particles that are then mixed with a sample preparation suspected to contain toxin in the wells of the microtitration plates. So, we have generated antibody against the particular foodborne toxin. It can be botulinum toxin, or it can be staphylococcal enterotoxin, it can even be Vibrio cholerae toxin, anything you talk about it. You would raise an antibody in the lab, monoclonal antibody, and this antibody is immobilized using latex particle. So, this is commercially available. There's many companies are there which produce these commercialized monoclonal antibodies which are suspended. So, depending upon what toxin you want to detect, you can buy the commercial kit. Now, this is mixed with the sample. You have the food sample which you suspect which contains the toxin. So, you mix your commercial antibody preparation using the food sample and then mix both in microtitration plates. The set RPLA can be used to detect Staphylococcus enterotoxins in a wide variety of food and to give a semi-quantitative result. So, if the specific toxin is present in the sample, you will see a diffused pattern in the bottom of the microtitration plate. In its absence, you will see a ring-like structure or button which can be visualized. For example, you have the VTEC RPLA latex agglutination test. This is a commercially available test against the verocytotoxin VT1 and VT2. The toxin can be, this can either be from the stool sample of a suspected patient, or you can use it to test the presence of the toxin in foodstuff. This is verotoxin is from E. coli toxin. So, E. coli isolates are recovered and identified from food or clinical samples using standard procedures. Then you have the thermoscientific company's BSET RPLA toxin detection kit. This uses the RPLA technology to find the Bacillus cereus enterotoxin. This Bacillus cereus enterotoxin actually causes diarrhea, gastrointestinal disease. This is a foodborne disease. So, if you want to recall the food, or you thought that you got the food poisoning by eating food from a specific source, to identify that it was the source, you can use this kit. So, you have a lot of commercially available kits which can detect the presence of the bacterial toxins present in the food. The next method is immunoimmobilization. This method was first developed for motile pathogens, that is pathogens which are capable of movement like Salmonella and E. coli 0157:H7. The motile cells and the antibody against flagellar antigen are applied on soft agar to enable them to diffuse in the same direction from opposite sides. So, what you do is you have the cells, the live organisms, then you have antibody against the flagellar antigen. These two are mixed together on soft agar. So, agar, since it is soft agar, the organisms can move and your antibody can also diffuse, and they will diffuse to the same direction from the opposite sides. So, when these two meet together, you will see a visible arc due to the immobilization, because the flagellar antibody will go and bind to the bacterial cells and stop them from moving. So, this binding would see a visible arc with which you can detect the presence of the microbe. Next comes the major topic, the enzyme immunoassay EIA, or otherwise we call it as the ELISA method. These methods were commercially developed and used to detect not only several foodborne pathogens and their toxins, but again this is also normally used in clinical conditions to detect other pathogens also, not foodborne pathogens alone. In the case of foodborne pathogens, we use the detection to find out the presence of Salmonella, Listeria monocytogenesis, Campylobacter jejuni, etc. The specific antibody is first allowed to bind on a solid surface. This solid surface can be the wall of the microtitration plate. So you use, for example, if I am saying detecting the presence of salmonella, I will use an antibody against salmonella. This antibody is bound to the surface of the microtitration plate. Now, then you add blocking agents. For example, bovine serum albumin, we add it to block the additional protein binding sites on the surface. So when the antibody is already added, it will have many sites, many protein binding sites. So you block those extra protein binding sites. Now you add the sample. The sample suspected of containing the antigen, which can either be the pathogen or the toxins, is prepared. Again, there is a specific volume, the conditions normally each kit has specific instructions, and this is added to the well and incubated for the antibody antigen reaction. So then you add the sample which has been prepared according to the instruction and you incubate it. After removing the unbound, so wherever the antigen antibody reaction has happened, they will go and bind. Then there would be some antigen which would be left unbound. So you have to remove it first. Then another antibody labeled with a specific enzyme is added. You add a second antibody after this. So you remove the unbound antigen, then add another antibody with which has been labeled with a specific enzyme. So what will happen is that this second antibody will go and bind to the first antibody, and this is now incubated for its binding to the antigen to form a sandwich. So you have a antibody, antigen, antibody, enzyme, the whole reaction. Again, the unbound enzyme linked antibody is then removed. So this sandwich complex is finally detected by adding a chromogenic substrate. You add a substance which highlights by color, specific for the enzyme which you have added towards the end. For example, 4-chloro-1-naphthol for peroxidase. This is incubated for a specific time, then adding an enzyme, and after a specific incubation period, you inactivate the enzyme using an exact inactivator. So what happens here is the intensity of the color development can then be measured to identify the presence of specific pathogen or toxin. So the type of color, the intensity of the color which develops will not only tell you the pathogen but also how concentration it is, because then the color will be intense. Instead of a chromogenic substrate, you can use a fluorogenic substrate also, for example 3-para-hydroxyphenyl propionic acid again for the substrate peroxidase, and then in such a case we measure the reaction using fluorimeter because it is a fluorescent substrate. Nowadays automated systems for ELISA are available. So this is a simple immunological reaction where the antigen antibody antigen enzyme complex will give a color reaction or a fluorescence emitting reaction to indicate the presence of either the bacterium or the bacterial toxins. This picture shows the presence. A capture antibody and a multi-well plate will immobilize the antigen of the interest. This antigen, as it shows in the picture, you have the capture antibody, you have the target protein here, you have the antibody here, finally the agent, you have the indicator here. So this detection would be based on the binding of the streptavidin and the biotin in this particular case example. Now, what are the advantages and disadvantages of this particular technique? ELISA is actually very sensitive, highly sensitive, and it is highly specific. So even when the antigen is in the presence of picogram, very less, it will be detected because of the specific, because we are using specific antibodies against a particular antigen. So it will be very specific, highly sensitive. Second, it is high throughput. We have commercial ELISA kits normally in a 96 well plate format, but you can also do it in 384 well plates, meaning that many number of samples can be isolated in a particular time. It is easy to perform. The protocols are easy to follow and involve very little hands-on time. Here what we are talking about is it sensitive, highly specific, very rapid, and since the instructions are very clear and you don't require much effort to learn and do the technique, you don't require much hands-on training to perform an ELISA. It is quantitative. It can determine the concentration of the antigen, since especially if you are using fluorogenic or chromogenic substrate, the intensity of the color or intensity of the fluorescence will tell you the concentration of the antigen which is present in the sample. and it is possible to test various sample types. You can test the serum, plasma, cellular tissue extracts, urine and saliva. So, this is a normal technique which is used not only for the clinical diagnosis of pathogens, but it can also be used to detect pathogens not only from the food, but you can also detect the presence, for example, if you suspect food poisoning, from that person's saliva, urine, you can detect the presence of the toxins concerned. Disadvantages: temporary readouts. Detection is based on enzyme substrate reaction. Therefore, readout must be obtained within a short time span, because enzyme reaction, substrate reaction is time bound. Once it reaches the maximum level, then it will slowly go down, because either the substrate is completely used up or enzyme becomes inactive, or both happens. So, it is a time bound experiment. Limited antigen information. Information limited to the amount of the presence of antigen in the sample. You are going to get an information only if the antigen is present or very less antigen, and you are not going to get much information regarding that. So, this becomes a disadvantage. So, this is a picture which gives you the different types, where finally you have the primary antibody conjugate here. Now, to the primary antibody conjugate, in this picture you are showing, in indirect ELISA we are talking about secondary antibody conjugate. You have a sandwich ELISA where you have a capture antibody, then you have the competitive ELISA. So, the basic technique of ELISA can be done in different manner, as the direct ELISA where you have a primary antibody conjugate, and indirect ELISA where you have a secondary antibody conjugate, a sandwich ELISA or a competitive ELISA. In the case of a direct ELISA, the antibody is immobilized on the surface of the, so you have to apply the antibody onto the multivalent plate, and this antibody is detected with a specific antibody specific for the antigen, and this is directly conjugated with HRP or the other direct. So, you have a direct enumeration detection of the particular pathogen concerned. In indirect ELISA, it is the antigen which is immobilized, not the antibody. So, antigen is immobilized on the surface of the plate. Then now you require a two-step process. One, you need to use a primary antibody specific for the antigen. So, you have the antigen which is immobilized, you use a primary antibody against the antigen, and then you use a secondary antibody which is labeled against the host species. So, you have a two-step process for the detection in the case of the indirect ELISA. This method can also be used to detect specific antibodies in serum sample by substituting the serum for the primary antibody. In sandwich ELISA, this is also called as sandwich immunoassay. This is commonly used format for the detection. It is a very commonly used immunoassay in many labs. You require two antibodies specific for two different epitopes of the antigen. So, the first requirement here is you require two antibodies, and these two antibodies are against different epitopes of the antigen concerned. So, this two antibodies we call as the matched antibody pairs. Now, one of these two antibodies is coated on the surface of the multivalent plate. So, you have already a matched pair of antibodies against the epitope of the antigen concerned. One among them is coated on the surface of the plate, and this is used as a capture antibody to facilitate, to help in the immobilization of the antigen. So, then the other antibody is conjugated and facilitates detection. So, the first antibody will capture the antigen, the second antibody will facilitate the detection of the antigen. So, we are using a pair, a matched pair of antibody against a specific antigen. In competitive ELISA, this is also called as inhibition ELISA. We measure the concentration of the antigen by detection of a signal interference, meaning the signal gets interrupted. The sample antigen competes with the reference antigen for binding to a specific amount of the label antibody. So, what happens is that you have a labeled antibody against the antigen and you have a reference antigen. So, your sample antigen, the sample which has been isolated either from the clinical sample or from the food sample, that will compete with the reference antigen for the same site on the labeled antibody. The reference antigen is pre-coated on the multivalent plate. Then the sample is pre-incubated with labeled antibody and added to the wells. Depending upon the amount of the antigen in the sample, more or less free antibodies will be available to bind to the reference antigen. So, it is concentration dependent. Depending upon the amount of the antigen available and free antibodies available, you will have the binding to the reference antigen. So, the more antigen there is in the sample, the less reference antigen will be detected. So, the signal will be weak. Some competitive ELISA kits use labeled antigen instead of labeled antibody. So, instead of using a labeled antibody, we can also use labeled antigen. So, it depends upon which kit you are going to use. These are commercially available kits. The labeled antigen and the sample antigen, unlabeled one, compete with the binding to the primary antibody. The lower the amount of antigen in the sample, the stronger will be the signal due to more labeled antigen in the well. Let us compare the different types of ELISA. The direct ELISA is a short protocol. It saves time and reagents. There is no cross reactivity. But the negative aspect of this is potentially high background. All the proteins which are present in the sample can react and can bind to the surface. In the case of indirect ELISA, secondary amplification, signal amplification is there. Several secondary antibodies will bind to the primary antibody. It is highly flexible. But disadvantage is it is very long protocol if you compare to the short term required for the direct ELISA, and you have the potential cross reactivity from secondary antibody. This is an indirect ELISA and a conventional typical direct ELISA method. Now, sandwich ELISA. Sandwich ELISA is highly specific. It involves 2 antibodies, but the disadvantage is you need to design 2 antibodies against the same target that recognize different epitopes, which will take time, and this sometimes might not work perfectly. In the case of competitive ELISA, this depends upon the base ELISA. This is required for or suitable for very small antigens concerned. And you have a newly developed technology which is called as the DAS ELISA, which can detect salmonella in chicken flocks. It can discriminate between infected poultry and uninfected poultry. Then you have another method which is called as GLISA, which can detect low up to 7.3 log CFU per grams, which is more sensitive than the other ELISA methods. And this GLISA method is commercially available for detecting Campylobacter in a rapid test. So, these are the, in a nutshell, these are the different immunological assays which can be used in the lab to detect the presence of the pathogens or the pathogen toxins from the food sample or from the clinical sample. The rest of the methods we will see in the next upcoming lecture. Thank you.